Welcome, oh, sorry. Welcome, everybody. Welcome to the Night of the Microbes. Thanks. I am Benny, I'm from Leipzig, and I will moderate this show tonight, and I will do it together with the wonderful Federica from Italy. Please welcome her with a massive applause. It seems that she does not come with this little applause. So please, do it better. Please welcome her with a massive applause.
Hello, everybody. Hello, thanks for such nice welcome, and thanks to be here tonight. We are hosting with very nice event that is inserted in the context of the 17th International Symposium on Microbial Ecology that this year is hosting more than 2,000 scientists from all over the world.
That's incredible.
Yeah.
And this year, the ISME visits Germany for the first time and is held in our wonderful city of Leipzig at the Neue Messe area. And the ISME is hosted by the Helmholtz Center for Environmental Research, also called UFZ.
Yeah. Since Leipzig is a very international and cultural city, the UFZ wanted to organize such event that is really the first of its kind in the long old story of ISME conference. And not only to show this such a huge conference, but also to draw the attention of Leipzig citizens about the microbiology and microbial ecology field that are very exciting and fascinating research fields.
And you all have made an incredible, smart decision to be here tonight. Definitely. We have 6 very talented speakers from all over the world, and they were selected by a prestigious committee. So you can be sure that you only have A-list presenters tonight. And they will talk about the fascinating life of microbes. By the way, Federica, do you have a favorite microbe?
Thanks for asking. Of course I do. You know, I'm Italian, and it's easy to imagine that my favorite microorganisms are the yeast.
The yeast?
Yes. You know why? Because the yeast, as you know and most of public knows, are responsible for the fermentation of the most beloved alcoholic beverage on the heart, wine and beer.
Sounds very reasonable, reasonable to me, Federica. But did you recognize it? I mean, you are from Italy, famous for its wine production. I'm from Germany, famous for the beer. It seems that I put the perfect combination on stage tonight.
But this has to be just a coincidence.
No, I'm not sure about this.
We will see. But now I'm curious, what is your favorite microorganism?
I like the yeast too, definitely. I mean, you just showed that's really important to all of us.
Yeah, I know.
But I also like the fact that there are so many microorganisms. Can you believe that in a handful of soil you will find billions of microorganisms?
Really? Wow, it's very impressive.
Here, you can even count. You can even count if you like.
No, thank you very much.
You sure you don't want to count?
No, you know, I'm busy now. Maybe later. Okay?
Later backstage?
Keep this.
Okay.
But it's really very impressive how microorganisms are really everywhere. And do you want to know last curiosity about microorganisms?
Of course.
Are you working with laptop?
Yes, I do it every day.
So you probably want to know, and most of you wants to know, that recently scientists have shown that there are 20,000 times
20,000?
more bacteria in the laptop keyboard than in the toilet seat.
Wow. That's incredible.
Yeah.
Maybe it seems that I have to clean rather the laptop instead of the toilet next time.
Yes, it sounds like a good idea. I would recommend you.
You see, microorganisms are everywhere, and they affect our daily life. So it's important to do research on these microorganisms, and moreover, to share this research. And the ISME is exactly about that researchers from all over the world come together and share their research. So do we have ISME participants, microbiologists in the audience tonight? Please raise your hand. Yeah, it's quite a majority. As expected, I would say. Yeah.
Such a surprise.
Do we also have non-microbiologists in the audience? Maybe a social science?
Woo!
Yeah.
That's very
All
good.
of you?
Yeah.
Nice that you're here.
Please keep this curiosity all over this show. It's important that we have non-microbiology.
And where are you from? People from Leipzig here? Yeah, through.
Quite a lot.
Germany, Europe,
Other country of yeah.
Asia. Yeah, all here.
Wow, we have a very
Hey.
well mixed and heterogeneous
Exactly.
publicum
We
today.
have a well-mixed audience, similar to our speakers tonight. We have speakers from the States, from Canada, from Switzerland, Netherlands, and also Germany. And I would like to make just one more point. So there's no competition tonight. There won't be a single winner. All of the speakers will be winners. So you don't have to vote with your applause for a speaker, but feel free to applause to the speaker at any time.
Yeah, and they are also very welcome, yeah? But words up, Benny, yeah? So we are here just to entertain you. The speakers will give very nice talk about their research field, and no winner, no competition. And I'm pretty sure that they will convince you that also scientists have a funny side, even if it's
Microscopic.
But now, let's give more details about the show.
Just a few words. So we have 2 sessions, each with 3 speakers. Each speaker will talk hopefully around 12 minutes. And between the sessions, we have a break of 15 minutes. In the break, you can get some new refreshments. And also the band Blaswerk will play. Blaswerk from Leipzig. And they will demonstrate that also scientists have a musical side.
This is important. And after the break, we will have other 3 speakers that will entertain you with their nice talk. And most importantly, after the show, you will have the possibility to get further refreshment and to get in contact with the speaker and ask some question in the case some curiosity arises during their talk. So now, let's get started.
It's time for our first speaker.
Yeah.
Please welcome Karen
Yeah, with
Lloyd.
a very massive applause. So she's Karen Lloyd. She's assistant professor at the University of Tennessee, and she works at the interface of marine biogeochemistry and microbiology, and she will talk this evening about the power of undiscovered microbes. But I have a curiosity. How did you know that there were undiscovered bacteria before discovering them?
Oh, God, that hurts my brain. I didn't.
Oh,
I didn't.
Such a pity. But then, let's get started.
Okay,
and,
thank you. Thanks for the introduction.
Stage is yours.
Yeah. Hi, everybody. I'm sorry, I'm not a comedian. Climate change is not something that's off in some hazy, distant future anymore, right? We're seeing the effects all around us. Most recently, in the long drought that Europe's been in, we've had some serious crop losses due to climate change. So I have a slide up of I pulled up, this is not Europe, this is South Africa. But climate change, as you may know, is caused by greenhouse gases like carbon dioxide, methane, and nitrous oxide. And microbes, you may or may not know, do more than just make us sick. They will make greenhouse gases, and some others of them will consume greenhouse gases. And crops would all wither up and die if they didn't get nutrients from the microbes that live at their roots and sustain them. And wastewater treatment plants all over in every city around the world are teeming with microbes that are working very hard to make our cities safe and to remediate our water system. So given this importance of environmental microbes, I want to tell you about a revolution that's been happening in environmental microbiology. And this revolution is that basically we discovered that our traditional techniques of looking at microbes had been missing quite a few microbes that turn out to be kind of important. So the analogy for this situation would be like ancient humans looking up at the night sky and trying to imagine how many stars they see. Some people estimate that this is 5,000, which seems like a lot of stars, but when humans developed the technology necessary to have really fancy telescopes, like the Hubble, now we know that there are more like 10 to the 25 stars in the universe. So this big leap forward happened because of technology. And I just forgot where I was. Right, in the 1880s, not too far from Leipzig, in Clausthal, did I say that right? Germany, Robert Koch invented the field of microbiology. And he did that when he got the very first bacterium in pure culture. So he grew it on its own in a petri dish. And it actually happened that the thing that he grew was anthrax, so it's really good that he didn't die. But that allowed us to have hundreds of years worth of discoveries that told us a lot about the microbes that are out there. But ironically, this ability to culture things is actually the thing that developed something that caused us to realize that culturing was missing a lot of things. And the thing that we learned was a neat little enzyme called Taq polymerase. This was isolated from a hot spring bacteria, and what it can do is make bajillion, million, zillion copies of almost perfect copies out of a single strand of DNA sequence. And this allowed us to go into the field. This technological advance allowed us to get sequences from out in nature and see what was there without depending on the things that will grow in lab. So to illustrate what this revolution is like, really, you can imagine that a biologist only knows about 4 species. Say we pretend that we know about us, we know about bunnies, we know about chickens, and we know about sharks. So this is actually kind of all you need to know, right? You've got adorable things, you've got food, you've got sharks for Shark Week on the Discovery Channel, so you're kind of all set. But imagine you had a technological advance that suddenly showed, oh my God, there's more than one kind of shark. This would make people really excited. And then your technological advance was so good that you could see all these different animals and your mind is being blown. Oh my God, there's fleas and lions. And then if your technological advance was so good that you can see really, really weird stuff that looks nothing like this, that just sort of blows your mind, like trees and grass and mushrooms. This is silly, right? We know all these things exist, but this is analogous to what we're experiencing right now in the field of microbiology. Except unfortunately, microbes are not this pretty, and it's hard to tell by looking at them what they do, so our revolution looks more like this. Aren't you excited to go to a talk about SPA 1033? I actually know a lot about that group, and I would love to tell you about it. But we've been trying to upgrade the names. It's been a point of discussion at this conference. You may notice some of them have gotten killed at this conference, but they're still up there. I had to give in my slides early. Some of them are named after Norse gods. So we're now stuck with a big question beyond just naming them, which is to figure out what they do and to figure out where we can find them. Are these novel, uncultivated things only present in exotic places like volcanoes? Or are they on my skin? Are they in the yard? And how many of them are there in this whole world? So it turns out that answering those questions is actually kind of tricky. Because remember I told you that we needed Taq polymerase to make a bunch of copies of DNA so that we could see it? So we can't just go out and tag these things like an albatross and estimate their population sizes. But one thing that we have going for us in this field is that for years, a lot of us have been putting these DNA sequences into public databases. So my collaborators and I decided to download all these DNA sequences from every environment, everything. Oceans, lakes, rivers, rain clouds, wastewater treatment plants, people's noses. And what we got were 1.14 million DNA sequences from all these environments, and they look obviously like that, sort of like that. So our job was to take each one of those sequences and figure out how similar it was to something that's known. So we compared them to this database of DNA sequences of known organisms. And this was great, and we could figure out how many changes there were, and then we can know how similar they were. But we hit a real computational wall. And the reason is that when you multiply those two numbers together, you get about 30 billion very computationally difficult things to do. So, this stalled for a while, and it was very sad, because I was like, I have this great idea, but I can't do it. But then I remembered at the University of Tennessee, we were very close to the Oak Ridge National Laboratory, and they have computers that look like that, which is way better than my desktop. And they let me put this equation in that computer. And do you know how long it took to do this thing that was completely intractable and stalling my research? Like 2 minutes. It was just done. I was happy, but I was kind of like, crap. All right, so, so that was done. And so now I'll tell you what we found about this thing. And I'll show it to you in the form of, I'll illustrate it with 3 different groups of microbes. So one group would be ones who are well known. These are the cultured microbes that we've seen before. And I will illustrate those with somebody that we can all recognize easily, I hope. We'll find out. Angela Merkel. And I've told this is the Merkel pose. You may love her, you may hate her, it doesn't matter. We all know who she is. So she's our stand-in for a cultivated microbe. So that would be 2 sequences that are very similar. Angela's the cultured microbes. So the next step out would be slightly different microbes. They're not in culture, but they're similar to what's in culture. And for that, I'll use a newborn baby. This would be, you know it's a human, so you know some basic stuff about it, but you don't know what her personality is going to be like. This is a new person you haven't met yet. Slightly different. And our third category would be microbes out in the environment that we don't know what the hell they are. They're space aliens. So those are our 3 possibilities for what microbes can be. And I'll show you how they break down in these different environments with pie charts, showing you the distribution of the sequences that we got. So first, let's go to this soil question. Does this stuff happen in soil? Well, it turns out that the tiniest pie puzzle piece is Angela, the cultured guys. She really isn't that much. Soil is mostly new babies and space aliens. It's mostly the uncultured stuff. And so what this means is that these novel microbes are not something that exists in strange places. They are literally underneath our feet. And we found similar things in oceans and lakes and rivers, the sequences were dominated by this new stuff. But you may say these are natural environments. Certainly if you went to a human-engineered place, we would have more common microbes that we're used to seeing. But even in wastewater treatment plants, they were still dominated by these new unknown microbes. And what this means is that these unknown microbes are probably really important for us as a human society. The one environment that was the true exception to this that kind of shocked us was our bodies. So if you look at the sequences that are present on skin and guts and hair, it's almost all Angela. So, and this is because we've just been spending hundreds of years cultivating things from humans, so we just did a really good job of it. Go microbiologists. Thousands of researchers right now, and many of them are here at ISME, are working on trying to figure out what these things do. And they're maybe thinking about ways to use them to mitigate drought problems, to make drought-resistant crops, or to create sustainable forms of agriculture, or to find new antibiotics, or to mitigate the effects of climate change. The answer for all those things could be here. And if you ask the title of my talk, The Power of the Uncultivated Microbes, well, Karen, what is the power of the uncultivated microbes? I would have to say, I don't know yet. Sorry. But I do know that they are very abundant in environments and systems that are very important to humans. And luckily, happily, we have the tools necessary to figure out what they're doing. And just like the discovery of Taq polymerase in a hot spring bacterium, it's hard to underestimate the power that that has had on human society. It absolutely revolutionized health science, agriculture, and genomic science and other things. Every uncultured microbe that's out there holds the possibility of a Taq polymerase-like discovery that will further revolutionize human society. And all we have to do is to look for it. Thank you.
Thanks, Karen.
It's terrifying.
Wow. Very energetic. Thanks, Karen, for such entertaining talk. It's good that you talk about Angela Merkel and you had the chancellor on your side. Very good strategy. So
now it's time for our next speaker. It's Alexandre Jusset from the Utrecht University, and he analyzes the interactions between plants and microorganisms. So, Alex, you traveled around the world, I read. You walked in China, in somewhere else. But you also were in Leipzig before, I read, so 15 years ago. So what do you think about the changes in Leipzig since this time? You're still.
Now, well, many things go, many things change, but one thing didn't change. When I came here 15 years ago, I thought, wow, this city is so fantastic. I want to stay here. Took another pass, and then I came here again, and I thought, wow, this city is so fantastic. I want to stay here. So maybe if you want, you could chat a bit with the people from who have said, maybe they have a job for me.
We can talk about this. Thank you, Alex. And so, I'm looking forward for your talk, entitled Microbial Gardeners for Pesticide-Free Agriculture. The stage is yours.
All right, thanks a lot. All right, today I will show you two things. First, we are screwed. Second one, microbes will save us. So, we all need to eat. I'm French, so I love eating, so in fact I do that all the time. But there is a problem. We have a big problem with food safety and security. So we have a growing human population that is growing and growing and growing, and this means also that we have more people to feed. So in this context, typically we have used two types of things, two types of strategies. One strategy is the most sensible one, just make new space, which is currently happening. So if you look at the forest in Indonesia, you find some relics of what was there to put oil palm. In similar places in South America. So it's a finite and not very sustainable way of improving food supply. But we still need to eat. So what can we do? We can improve the yield. And for that, we have been massively building on extremely intensive agricultural strategies. And these strategies have increased the yield by hectare by a factor 10 in the last 10 years on the short term. Because now, the cost of these strategies is becoming so overwhelming that we have to think about new way to save the world and feed the world. And mainly so, massive application of fertilizer and pesticide has led a widespread degradation of soils, killed insects, and killed pretty much everything we needed, and generated a lot of pollution problems. What are we going to do? Well, luckily, well, that's not lucky, that's where we are heading. But there is a way to avoid that. Because you wasted this good soil here. This is a treasure. This is a treasure that may help us to eat in the future. I would not waste it that easily. Because one of the major problems we are facing now are plant disease. Plant diseases are causing up to 20% of yield loss worldwide, which means essentially the population of China or India doesn't have to eat, what they do, but because of pathogen, to put it in a system. So, and currently, if we want to fight this pathogen, we require massive amount of pesticides that will kill bees, cause a lot of problem to workers, pollute the groundwater. We cannot do that anymore. Otherwise, our soil is really over in a very short time. Luckily, we are not alone. Plants are not alone. You have maybe heard of the concept of probiotic microbes. If you have a kind of upset stomach, you can eat them, and at least according to the advertisement, it will work. It will restore your belly, your health, everything. It may or not work by human, but what we know is that plants carry with themselves a microbial pharmacy. In these few grams of soil that I carelessly threw away, a plant will be able to find out the microbes that keep it healthy. And if you look at a plant, if you pull a plant out of the ground and you look at the roots, you will find that this plant is associated with billions of microbes. It is covered by a slime. And these microbes can be extremely important for the plant. And you even have a kind of mutualistic interaction between plants and microbes. The plants feed the microbes. And some microbes, in return, produce several antibiotics that naturally keep the plant healthy. You can see an example here of, on the Petri dish, you have in the center a very nasty pathogen, a fungus that eats up roots, and here, these bacteria produce antibiotics that prevents plant growth. As a result, these bacteria may function as a shield that will ward off pathogens and keep plants healthy. A couple of years ago, I was with my good friend and colleague John Wayne from Nanjing. We were sitting not around a piece of dirt, but around a delicious plate of feichang, and thinking about the world and how to keep on working together. Suddenly, his phone rings. It was a farmer, Jiang. And he calls, Professor, I have a problem. Please help me. You're so smart and brilliant. I need your help because all my plants died. I don't know what to do. My whole harvest. So then we took the next taxi. We went to his field. And indeed, all the plants were dead. They were infected by a Ralstonia, terrible disease that is killing more than 200 types of plants. It's a bacteria. It lives in the soil. Then it grows on the roots, enters the roots, and then clogs everything and kills the plants within days. There is no cure. So we went to him, and then we thought, maybe this kind of concept of microbial shield could be indeed could serve as kind of a soil health immunization technology that we could put to keep the plants healthy. And we went back to the lab, and we thought, maybe Pseudomonas is a nice candidate. So it's a nice bacteria that grows on the plants, is known to produce several antibiotics, and it's easy to grow, to produce on a biotechnological scale. So what we did was something very simple. We know, okay, we have this microbial shield, in theory. However, if you have a poorly managed soil, this shield will be broken. Because by having a bad management, you killed all the good microbes. So we said, maybe you can restore them. So we took the plants of healthy plants, the root of healthy plants, we shake them, took bacteria, selected the most promising one, and then we went back to the farmers, and we added these microbes on small tomato plants. So that they get colonized by this shield, you restore the shield before putting them in the soil full of pathogen. With the idea that these nice microbes, if we selected them correctly, they will grow on the roots, form this kind of slime around the roots, so that if you have a pathogen that wants to enter, it will have a very hard time, because first, there is no space. It has to enter the roots, but all the roots are covered with microbes. In addition, these microbes will produce massive amounts of antibiotics. So as soon as the pathogen comes, well, it dies. It was quite a challenge to find the right microbes, because that's the ideal case, but you need also to have good microbes. But after a couple of trial and errors, we found a perfect mix of microbes that was extremely good at growing on plants. We went back to the farmers, gave them this bacteria, he put them on the roots, and that's the result. Even we didn't believe it. None of the plants was ill. And this means that by having a kind of easy screening and relatively easy screening, you can really replace pesticides. This is one of the many diseases that are threatening yields. And instead of throwing tons and tons of toxic compounds on your field, you can just add locally on the roots some beneficial microbes that would naturally produce these microbes for you. And with that, you have an extremely powerful way of securing food supply without destroying the environment. And as a result, so now we have kind of a very good basis. And now we are developing further strategies. So we are teaming up with companies who can really produce the right bacteria. We are doing still a lot of research. This is a case study that we're extremely lucky to solve. But we are still doing a lot of research now trying to understand what makes these beneficial microbes happy so that we can develop further strategies. We are also kind of trying to develop microbial communities that are extremely good in different environments. And more than ever, we are fighting with regulations because currently in Europe, it is impossible to develop such product. It is so good as forbidden. If you have this type of product, you have to show that it is biodegradable and doesn't kill bees. It has a bit outdated regulation, but we are still trying to cope with that. In China, there are already solutions. They are 10 years more in advance than we are. But here, it's also something that you can help. That if you are, as a citizen, and especially if you are involved in politics, I think it is a big misunderstanding about microbes. There are still a kind of a big clash between how bureaucrats see microbes, administration sees microbes, and how they are. And with your contribution as a citizen, as a scientist, you can advise your representatives to promote a more friendly regulation so that the farmers in the future can have a sustainable and safe and productive agriculture. So if you are interested in a nerdy person, you can read these papers or send me an email. I would be very happy to answer you. And of course, I presented everything in a very romantic way, but this is a teamwork. We have several persons who are involved, a couple of them in the room. So Houdier, who is doing a PhD in Nanjing, and Utrecht, stand up. And Wei Zhong, who is who have been doing a fantastic work. And also, this is a big teamwork, and I would like to thank all my colleagues in Utrecht and in Nanjing for the fantastic teamwork. This is our team, and together we will save the world. Thanks.
Thank you, Alex, for this inspiring talk. Maybe we have a chance to survive if you follow your work. And it's time for our third speaker.
Yes, she's Mary Beth Lee. She's professor of microbiology at the Institute of Arctic Biology in Alaska. And she did the NASA Space Training Program. Is that right?
That's true.
Yeah, so did you really go to space?
I did, actually.
Oh, really?
This space right here. Yeah.
But you are also a very good dancer and choreographer. So are you going to dance this night?
Only if you lead.
Okay, so the stage is yours.
Okay, thank you very much. So this evening, I'd like to take you on a journey into the microbial world as seen through the eyes of artists. These are artists that have worked together closely with me and over 30 other scientists who have a passion for the role that microorganisms play in the environment. And you know by now that I'm a microbiologist, but I'm also someone who has a background in the arts. And so I've had a long-standing interest in answering this question. And I've been doing lots of experiments over the years to answer this question together collaboratively with artists and scientists in Alaska. And over the last 12 years, we've collaborated together to produce various art exhibits and live performances with dance and readings and scientific presentations for the public on all kinds of themes like climate change and wildfire, and my favorite theme of all, microbes, which is what I'll tell you about tonight. In this project, 13 competitively selected artists went on a journey into the microbial world that began with lectures and lab activities, but we spent a lot of time then doing field trips. And this is us on a field trip on our way out to discover microbes in their native habitat. And we explored their microbes and their lives in the boreal forest near Fairbanks, Alaska. We went north across the Arctic Circle to Toolik Field Station to discover Arctic microorganisms, meeting with all kinds of scientists, learning about what they do, and taking samples alongside them, which the artists could then take back into their own homes and laboratories. They all had their own microscopes to discover on their own. And they became their own independent microbiologists and developed their own research interests. And then developed, much like a scientist, methods and creative original works on the themes of the microbial world that they discovered. And the result was an art exhibit that was a collective exhibit with these 13 artists that's been touring around Alaska for the last year and a half and also to Finland, called Microbial Worlds. And lots of different aspects of microorganisms inspired the artists, including just their basic visual beauty. When you look in a microscope, they have incredible variety of shapes and sizes and arrangements, like in these wood carvings by Sarah Tabbert. The artists were also so amazed to learn how widespread they are. For example, these threads of fungal cells that permeate the soils and constitute the majority of biomass, we call it, in soil, along with bacteria. And these fungi partner closely with plants, and the plants rely on them in order to survive. And of course, periodically, these fungi will send up a little mushroom that might be delicious or beautiful or make us see funny things. So these are the things that we see, but they're really the tip of the iceberg of all the fungi that live in the soil of the boreal forest and all soils around the world. We also discovered microbes in places where the artists didn't expect to find them. For example, in permafrost. So this is a core sample of permafrost that we took near our university in Alaska. And permafrost is defined as soil that's frozen for 2 or more years. But really, much permafrost in the world has been frozen for thousands of years or even millions of years. And what we've learned as science is that no matter how old that permafrost is, it contains living microorganisms, because if you bring it into the lab, you can culture them. And here are some examples of colonies of bacterial and fungal cells that have grown out of ancient permafrost that were provided by a scientist to one of our artists. This is a detailed shot of this larger piece highlighting the diversity of microbes and how they bloom as permafrost thaws. It's really incredible. And this permafrost sample that yielded these microbes was taken from the permafrost tunnel, which we have in Fairbanks in Alaska. You can just walk into a hillside and go back in time to the Pleistocene, when the soil froze, back when mastodons and woolly mammoths walked around Alaska. And we can find mastodon bones sticking out of the wall, but what's even cooler is that we can find microbes and also plants. So what Tom is showing here are plant roots and leaves that have been essentially freeze-dried in the permafrost from 10 to 40,000 years ago. And what this means is that when the microbes awaken, when the permafrost thaws, they have plenty of food to eat right there because they love to eat plant material. So it might be in the form of these large roots and leaves or maybe in smaller particles, but there's lots of organic matter in this permafrost. And so when microbes thaw and come to life, they start to eat and they start to belch. And this is what you can see in frozen lakes in the fall. This is the exhalations, if you will, of microorganisms from typically the thawing permafrost under a lake in northern regions that are releasing not only carbon dioxide, like we do when we exhale, but also methane. Because when microorganisms eat plant matter without oxygen around, like at the bottom of a lake, they produce a lot of methane. And this is a very concerning thing, as we'll talk about in a minute. But it's also very beautiful. And so this struck the artists, Ree Nankara, who's Alaska's leading textile art quilt designer, who made hand-dyed and hand-sewn beautiful quilts, including this one called Deceptive Beauty, which really touches at the fact that there's actually danger underlying this beauty that we see in frozen bubbles in the ice. And as we zoom in closer to her art, we can see that there's a complex community of microorganisms here working together to eat that plant material and to release CO2 and methane. She also has a lot of images in this quilt of fire. And this actually refers to our popular pastime in Alaska of lighting methane bubbles on fire. We love to do this. You go out and poke a hole with a match and boom. And this is scientist Katie Walter, who's an expert in predicting the amount of methane that's going to come out of permafrost as it thaws in the northern regions. And this is a concerning thing. So permafrost is a pan-Arctic phenomenon. We have it all around Canada, Alaska, and a great deal in Siberia. But this permafrost is disappearing quickly as the climate warms. So projections of in the next 100 years are that up to 90% of this permafrost may thaw. And that's very concerning when you realize how much food there is in there for microbes to produce methane. This is a problem because methane is a very potent greenhouse gas. We know that CO2, carbon dioxide, is a greenhouse gas that warms the planet, but methane is 25 times stronger at trapping the sun's heat. And so what this produces is a positive feedback loop when permafrost thaws. So we know our climate is warming, increasing the temperature, causing permafrost to thaw. That's causing the microbes to wake up and eat and belch carbon dioxide and methane into the atmosphere, which further warms the Earth. melting more permafrost, releasing more methane. It's a positive feedback. So we're in a very dire situation when it comes to climate change when you take into account the permafrost that has yet to thaw. But microbial decomposition is not always a bad thing. Actually, if it didn't happen, we would be in big trouble, right? We wouldn't have nutrients for our crops to grow with, and we would be up to our ears in leaves and sticks that have fallen out of the forest. So this idea of decomposition was something that Stephanie Dixon explored deeply in her series of works, of multimedia works. And one of the things that's amazing about microbes is their ability to eat things that we can't eat and to digest things like complex plant fibers. That the only reason we can digest some of them is because microbes are there to do it for us. And of course, cattle have the power of microbes in their stomachs to help them eat things like leaves and hay or straw. So Stephanie Dixon is an artist who came along with me and my research team to northern Finland for 3 field seasons. She did all the science alongside us, and also then we converted the lab to her art space and we helped her with her art projects. She's a costume designer and built dresses that she had us wear during photo and video shoots. And she was very deeply inspired and actually incorporated scientific methods into her artwork, which I think was an interesting advance that she made in this project. So our research project was to understand what microbes are involved in decomposing plant litter in this boreal forest, and also what factors control that. And we used standard methods, which many scientists do for this. You take leaves, you put them in nylon bags, in this case pantyhose, nylon stockings. You can set them out on the ground or bury them and dig them up every period of time in order to measure the amount of mass loss, so how much of that the microbes ate. And then we can also analyze the DNA, as you've heard about earlier, to identify the microbes that are involved. And so Stephanie actually incorporated this process into her artistic creative process as well by creating dresses with photo shoots. So here's a fresh dress with a very overt reference to microorganisms. But then she took it one step further and buried those dresses and let them decompose for 1 and 2 years, dug them up, had us wear them again. By the way, that's a very cold experience in September in the Arctic, and did repeat photo shoots. So here's an example of a dress that was laid out on the surface for 1 year that she had me crawl into as it was, including all the biting insects inside. And she also got so excited that she started to bury us as well. So this is my postdoc, who was a very good sport and was buried alive in the tundra. And once she dug us up and our dresses, then what was really fascinating to see was the amazing effects the microbes had on the textiles. So here you can see some fungal colonies. The fungi have released enzymes that break down the plant matter that have bleached the fabric. And what we did was to then take DNA from this fabric and analyze it as a live experience for the people who came to the art show. Using an incredible new technology, this is a DNA sequencer that's just handheld. It's called a MinION. One of the newest generations of DNA sequencing technology. So the audiences who came to see the exhibit could also see in real time the discovery of all of the microbes that were decomposing these dresses. And the results look something like this. We have lots of bacteria. That was 73% of what was decomposing the dresses. And then things like fungi and other organisms were a smaller fraction of that, including many things that were as yet to be identified, as you've heard about already. So we know that microbes are really good at decomposing leaves. What they're really bad at decomposing is toothbrushes. And this was a concept that really inspired artist Margot Klaas to create a series of artist box constructions using found plastic that she'd found on the shoreline of the ocean in the Pacific Northwest. And I think we've all heard about this problem and how serious it is that we've released a lot of plastics in the environment that are just not decomposing. So Margot created a whole set of these beautiful constructions that are also at the same time tragic, I would say, with pieces of found plastic, including beautiful handmade artist books that inform the audience about the issue, and also how to reduce their use of plastic and reuse and recycle. And there was also a hopeful element tucked in here in the form of this book, which explains an exciting new scientific discovery that was made by a group of researchers from Stanford and from China that discovered that there are microbes that can break down plastic, and they live in the guts of mealworms who like to eat styrofoam. And so, amazingly, there are microorganisms that can help us with this problem. We're just starting to find them. It's a tiny step forward towards solving the plastic problem. But what Margot did was to take the scientific information from this paper and recreate the graphics and add some commentary that was much more digestible, if you will, for the public than the original scientific paper that's really meant for specialists. And so this raises the question, I think, of what is accessible to the public? What did the public get out of this experience? So we did have really high numbers of people who came to see the show. That's one way of measuring impacts. But did it really teach people anything? Did it change their attitudes? We've been doing research on this in collaboration with social scientists through audience surveys, and finding that the majority of people who attend these art events reported an increased knowledge about the role of microbes in ecosystem, also an exciting increased motivation to learn more. This also has implications in education for combining the arts and sciences together for students. And knowledge of effects of things like ecosystems and how they function, how human activities are affecting that, and just a general increased appreciation for science. And this is not something that I've invented by any stretch. I think the fact that we have these positive outcomes is one of the reasons there's been a groundswell of arts, humanities, and science collaborative activities across the US and also across Europe. I show this figure because my collaborator has assessed particularly the arts and humanities research that's been done across US ecological research sites, and there's now over 100 with active programs of all different sorts involving lots of different media. And there are many exciting things happening in Europe on the front of art and science interaction as well that I think we'd like to look more at, and we'd like to network together and make more advances on this front. We've published a paper examining the impacts of arts and humanities integration on audiences, and found that what it has the power to do is to strengthen relationships between scholars and the public and the natural world. Of course, inspire and cultivate empathy for the natural world among people that are involved in these projects and the audiences that come to see them. And also, really importantly, spark awareness that can enable pro-environmental behavior, because as we all know, right now we're facing a major crisis, the grandest challenge perhaps that humanity has ever faced, and that's climate change, which is causing all sorts of havoc on the earth: wildfires, coastal erosion, loss of diversity, drought and famine. And this is a problem that science alone cannot solve. Science is really good at doing a lot of things. Science is awesome, and I love science. It can tell us what we know about the world, how it works, and where it's going, what its future looks like. Science can do this, but what we have to do as a society in order to decide what we're going to do and to solve a problem is to go beyond just simply the knowledge of what's happening, and combine that with our feelings, our values as a society, our aspirations of what we want to be, who we want to be, and how we want to take care of our earth. This is really in the domain of the arts and humanities, including philosophy, environmental ethics. And we have to put these two pieces together before we can decide as a society, what are we going to do about this problem? What should we do? So this also has to engage the public and society and policymakers. So what we really need is very close interaction and exchange across these disciplines and with the public and with society. So coming back to why I do this experiment in art and science, it's not only just because I think it's fun, which it is, but it's also because I think it could have a higher purpose potentially. And that's, in the words of environmental ethicist Michael Nelson, get us a little bit closer to harnessing the full power of the human imagination to tackle this grand challenge that we face right now, to reconnect the arts, humanities, sciences with society by working together collaboratively. So with that, I'd like to thank you very much for your attention. Thank you.
So thank you, Mary Beth, for such inspiring talk, even if I really miss you dancing around. But thank you, it was very inspiring.
So, ladies and gentlemen, this was the first session of Night of the Microbes. We hope you enjoyed our first 3 wonderful speakers. And now we have go for it. And now we have a little break, about 20 minutes. You can get some further refreshments and hear some music.
Yes, we will have the band Blasberg from the city of Leipzig that will play for us. And we have also some UFZ scientists that are actually playing in this band. So as we have just heard, we can show that actually music and science don't exclude each other, but actually are just different forms of arts. So enjoy the break, enjoy the band, and see you in 20 minutes.
Good job, Federica.
Seid ihr auf On?
Ja.
Ja. Nein.
Nein, wir sind doch.
Doch.
Langsam
Sind wir auch
kommt
hier?
was.
Wir sind noch nicht ganz auf dem Monitor.
Noch nicht auf den Monitoren zu hören,
dran.
aber für Sie zu
Er
hören.
arbeitet dran.
Genau. Langsam.
1, 2. Ja, jetzt wieder.
1, 2, 1, 2.
Wir sind da. Blasberg. This is my lovely colleague Yola. I'm Christiane, and we have some more songs prepared for you. Next up is Big Spender. You might know it in the version from Shirley Bassey. The minute you walked in the joint, I could see you were a man of distinction, a real big spender, good looking, so refined. Say, would you like to know what's going on in my mind? So let me get right to the point. I don't pop my cork for every guy I see. To have fun, fun, fun. How's about a few loves, loves? I can show you a good time. Wouldn't you like to have fun, fun, fun? How's about a few loves, loves? Let me show you a good time. Hey, big spender. The minute you walked in the joint, I could see you were a man of distinction, a real big spender, good looking, so refined. Say, would you like to know what's going on in my mind? So let me get right to the point. I don't pop my cork for every guy I see.
Christiane. Following song is a song you probably all know. It also exists in German, but we'll make the English version for you tonight. It's Mac the Knife. Oh, the shark has pretty teeth, dear, and he shows them pearly white. Just a jackknife was my key, dear, and he keeps them out of sight. When the shark Look out, old Mac, he is back. Mac the knife. Thank you. Next song, I asked Christiane to come back for Making Whoopee.
Another bright, another June, another sunny honeymoon, another reason, another season for making whoopee. A lot of shoes, a lot of price. The groom is nervous, the answer's twice. It's really killing that he's so willing to make it whoopee. Now picture little love nest down where the roses bloom. Picture the same sweet love nest. Think what a year can bring. He's washing dishes and baby clothes. He's so ambitious, he even sews. But don't forget, folks, it's what you get, folks, for making whoopee.
Another bright, another June, another sunny honeymoon, another season, another reason for making whoopee. A lot of shoes, a lot of rice. The groom is nervous, he answers twice. It's really killing that he's so willing for making whoopee. Picture the same sweet loveliness down where the roses cling. Picture the same sweet loveliness down where the year can bring. He's washing dishes and baby clothes. He's so ambitious, he's so don't get folks. That's what you get, folks, for making whoopee. 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 Making whoopee.
Thank you.
Thank you. This is the last song before we leave you back to your microbes and microorganisms. But this last song will blow your mind because this is Ain't No Mountain High Enough.
Ain't no microbe.
Ain't no microbe high enough.
Listen, baby, ain't no mountain high, ain't no valley low, ain't no river told you you could always count on me, darling. From that day on, I made a vow I'd be there when you want me someday, somehow. Baby, there ain't no mountain high enough, ain't no valley low enough, ain't no river wide enough to keep me from getting to you, babe. Oh no, darling, no wind, no rain, oh winter cold can't stop me, baby. No, baby, 'cause you're ever in trouble, I'll be there on the double. Just send for me, oh baby. My love is alive, way down in my heart. So go where your miles apart. If you ever need a helping hand, I'll be there on the double as fast as I can. Ain't no mountain high enough, ain't no valley low enough, ain't no river wide enough to keep me from getting to you, babe. Ain't no battle mountain, ain't no battle valley, ain't no river to keep me from getting to you, babe. Don't you know that there ain't no mountain high enough?
Thank you.
Thank you so
Thank
much.
you.
Have a great
Glaswerk
evening.
Leipzig. Thank you. Cool, sehr schön. Thanks. Have
Welcome back, ladies and gentlemen. Please grab your beverages and take a seat. You're going to continue our great show, Night of the Microbes.
So we hope you really enjoyed the music from the band, and now you are very ready and energetic yet for the second session. So let's welcome on the stage our first speaker of the second session. So Josh Neuefeld is professor in microbiology and microbial ecology at the University of Waterloo, and he's studying how microbial community interact with each other and the environment. So
oh, thank you.
So I'm curious, you have a German subtitle. Sprichst du Deutsch?
I'm sorry?
Nothing. Let's come back to English. Yeah?
Okay, that's a good
and
idea.
Let's talk about human skin microbiome. Stage is yours.
Okay, thank you. Okay. Please don't move, freeze. I'd like you to look down slowly at your hands. Don't move. If your hand is holding or touching anything at all, I want you to lift it slowly where I can see them up in the air. That's your right, maybe. If your other hand was touching something, I want that in the air as well. Okay. Even if they were touching each other in the air. Now, I want you to slowly pass the beers towards the front. But seriously, you can put them down. Thank you. But the point is, oh, you're a good man. Thank you. Cheers. You can have that back. Thank you. The point was, you were not thinking about your hands before I started that process. But from tonight onwards, I think you will think very differently about your hands every time you think of them, anytime you touch anything, because there's more to just you. There are your microbes that are on your body as well. And I want to introduce you to your skin microbes. Etwas hautnah erleben. That means up close and personal, more or less. So that's what I want to talk to you about tonight. I want to also begin by taking you back in time to Thursday. Thursday morning, I got up, I had a doctor's appointment, and so I drove there. Hand on the steering wheel, other hands were on the steering wheel before, for months and months before, and I finally arrived at the doctor's office. I had to go to the bathroom, I had to go to the toilet, so I put my hand on that door handle, which has been touched by hundreds of patients over the past year. After that, I turned on the light. I did some things. I did this, I washed my hands, talked to the doctor, and I'm not taking pictures of that situation, but the next thing that happened, I was at work. I was in the office, and I was thinking. I was adjusting. I was resting. I was eating, chewing, whatever. So what's going on in all of these images here? What's going on is this. We're zooming in a little bit here. And this is the habitat. This is the environment of the skin. It's all different. There's lots of different components to that skin surface, and you can see the microorganisms on the surface of the skin. It's not unrealistic. And if you were to take the skin from me and hold it up like a sheet, you'd have 2 meters squared of skin. And on that surface of skin, if you were to shake it off onto the floor, what you would find is 1 trillion microbial cells. Bacteria and archaea and fungus, all kinds of microorganisms that inhabit that skin, hundreds of species, maybe even thousands of species there. So that may be disturbing to some to think about, but at the same time, almost all of these organisms pose no harm to us whatsoever. In fact, many of them are very important for our health, and some of them are important for teaching us about what disease looks like and what normal looks like. So for that reason, it's important to study the normal microorganisms that are a part of us so we understand what disease looks like and how to fight those other organisms, the pathogens, the bad ones. Okay, so what is normal? Well, this is normal. This is a cross-section of the skin, kind of sliced in half there. So you see the sweat glands down below, which puts some oils up on the surface, a little bit of food there. You can see the microbes on the surface of the skin right here. And this, as an environment, a habitat for organisms, is not an easy one. These organisms have to contend with low temperature, a little bit cooler than the rest of our body, low pH, which means it's more acidic, more acid, low moisture. Microbes love moisture, but there's very little of it on the surface. There's also not very much food available, and it's also harsh because there's salt present there and antimicrobial compounds, things that our body produces that tries to avoid other organisms that might cause disease being found on our skin and causing infection. So that's what it looks like as an environment, and as a microbial ecologist, I study environments. Normally, I study soils and water, and as I approach skin for the research I want to tell you about, I don't see a big difference between these types of habitats. And as we sample from soil with a shovel or water with a filter like this, it's not that different from taking a sterile swab and rubbing that across the surface of the skin. So that's how we sample that particular habitat. Okay, so that's what the sampling looks like. As we begin, we know a few things about those skin microbes. We know that you have a unique collection of microbes, which looks a little bit like this in these cartoons, and they vary depending on what part of the body you might sample. So the oily regions may look a little bit different than the moist regions or the dry regions. And over the course of a year, it's relatively stable. It doesn't change too much, but there are a number of factors that can influence the composition of all those different locations. Okay, so these are some of the factors shown here, like temperature, if you're warm, for example, or the things that you're in contact with. Okay, so we know that, but and so in that way, you are a little bit special, like a snowflake. You have your own collection. It's different than the person beside you. It's your special you, that characteristic that you have, those unique microbial communities. So we know that. But what we didn't know very much about is how that unique community that we share as humans compares to other organisms as well, other mammals that are closely related to us evolutionarily. So my student, former student, who was doing her master's research, that's Ashley Ross, she was curious how the human microbiome might compare to these other mammals here. So she was busy. For the first year, she was sampling from 200 individuals. Multiple samples for every individual means hundreds of samples across 40 species and 10 mammalian orders. And then what happened? What happened next is, this is how the sampling worked, by the way. She was sampling from the back and the front and this moist region right here, the thighs, left and right side of all of these animals. At the same time, she collected the exact same samples from the human participants in this study, but as you can tell right here, we sampled a lot more from the human participants. And so the other samples will become important in just a second. But for now, keep in mind, 3 samples per organism, including the humans. That's what I'm going to show you the results from. How do we look compared to other mammals? Okay. All right, so this is what happens. You take the skin swab, you cut off the tip of it, and you extract the DNA from each swab, and then you target by sequencing, like the DNA sequencing approach we talked about before, a key gene that's found in all of life. And so we can collect that gene from all the organisms that are present, and we analyze that. And so what the data looks like, the results look like this when we collect it. You have, for one swab sample, microbe 1, here's an organism DNA sequence, microbe 2, here's another DNA sequence, and that goes all the way down for thousands of sequences. And then we want to compare sample 1 to sample 2, and 1 to 3, and 2 to 3, and every pairwise comparison possible. And then, yes, Ismi, I'm about to show an ordination. This is what it looks like when you compare all the samples to each other. This type of a visualization is a 2-dimensional visualization. All of the swab samples are here. So each circle is a skin sample. And if 2 circles are close together, like two at the top are close together, they have a similar microbial community. Okay, so let's orient ourselves now within our actual data, which is this, and I'll add on the color codes, the orders. You don't need to read the links between the orders and the colors. I'll just highlight a few that tend to be grouping close together. So we have some kangaroos down here, cows over here, bat order over here, some primates over here, rodents up here in a group. Okay, so on this side here, in this big cloud of sequence data from different samples, we have all of the mammalian orders. You might notice that there's sort of something missing here, or at least there's something sticking out to the side, and you can probably guess already what you're looking at. Those are the humans, all the humans over to the side. So in this way, the humans are distinct from all other mammals, except some of you who are looking very carefully notice that there's a few carnivores sneaking into the humans, and so we can add those as well. These are the carnivores that we share our homes with, and these particular carnivores don't go outside. They look a lot more like we do. For whatever, a variety of potential reasons here, hard to say, but the dogs and cats that spend time outside look like the rest of the mammalian cloud. Okay, so in that way, we are unique as a species. And on top of that, so I can simplify it here. Humans look like this, non-human mammals look like that, and this is based on what you can collect with a swab. So they're distinct in terms of composition. But if you look at the numbers of species as well, we call that diversity. If you sort of look at a graph where you have diversity, higher you go, more diverse, and all of the orders that we sampled, and here's the humans right here, I'm pointing to it with a swab as well, lower than all of the other mammalian orders. So it looks as though humans have also a subset, a smaller set of the diversity of the other mammals. So I'm sure you're thinking already it must be because of we're washing, it's more hygienic. And that may be true. That may be what results in us sampling a different set of skin microbes. So, but bottom line is though, we as a species, with what you can remove from the surface of our skin, is quite different than all other mammals. So it's quite special. We as a species are like snowflakes. We're unique. Okay, so that's the mammalian skin microbiome survey and where humans fit in relation to the others. But I kind of hinted that the humans had a lot more samples, and that was a really fun embedded experiment, something that we had also within the data. And so this was an interesting collection of humans. These were 10 couples, and they were all romantically involved, cohabiting, heterosexual couples. So our question was whether, by sequencing all the skin microbes from all these body parts, we might be able to put the couples back together just based on the skin microbes. Can you find your partner this way? So, in doing this, and with all these samples, what body parts do you think might be most appropriate? Before you guess, I don't really want to take any guesses, but I'm going to suggest that we just choose a random site from the body and look at the data from the random site here, which is the inner thighs. And when you look at the inner thighs, as shown here, we're not seeing that couples are grouping together. Instead, what we see, same kind of data display, you have the left and right inner thigh, left and right inner thigh. Each individual has a unique connected left and right inner thigh when we had the two samples. The males are all on the left, the females are all on the right. So here what we see is biological sex dictates what microbes are found in the inner thighs. Left and right side, you look like yourself. So that's not useful for putting couples back together, but if we look at another body part, and you will never guess, the other body part is the bottom of your feet. The bottom of your feet, and this is a great example, couple one, female, couple one, male, and here the left and right feet are very close together. They look like themselves, but they're also grouping together. And that tends to be true for the other couples as well. They're very close together. So, in that way, the answer to the question of whether we can put couples back together or if we have a random grouping when we teach a computer, here's the data, figure it out, The answer is that 90% of the time, roughly, you can put the couples back together again, which was much better than possible by chance alone. And it's most apparent by sampling the feet. For whatever reason, and you can imagine what those reasons might be, but as you move around your house, and as you, with bare feet, share those same surfaces, and by the way, this is the morning of heading onto a plane to come to Leipzig. This was me getting out of the shower and walking on the floor that the rest of my family shares with me, and now I look a little bit more like them, and they presumably look more like me. So that may be the reason. That's the best guess that we can come up with for that. I love what the media did with this story. I love the feet, always the feet, the stinky feet that unite us with our loved ones. And I really like the choice of image that Popular Science. It's an intimate display of bacteria swapping. Go figure. So, you, as a species, are a snowflake. You are special, and yes, you as an individual are also special, but I think it's quite appropriate that you also share some of that uniqueness, some of what makes you you with your loved ones. So the conclusion is, take-home messages here that humans are unique among mammals, your skin microbes are unique to you, and your skin microbes are shared with those you love. And next time you take a shower, you're not just washing microbes off. You're maybe adding some on as well. And the next time you try something new with your partner, remember that your bacteria are having a good time as well. And thank you. Thanks. Okay.
Very, very nice talk, Josh. I still feel undecided if I should take a shower or not this evening. I will think about.
Yeah, me too. Our next speaker is scientist at the Helmholtz Center for Environmental Research. So she's a local. Please welcome Anja Worrich. Welcome, Anja. So as a local, could you already give your colleague speakers, some speaker colleagues, some advices for the after-show party tonight?
Well, I think the best after-show party will happen directly here in the Werk 2, so stay until it's finished.
Yeah, this sounds like a good option. Anja, I'm looking forward for your talk, a fairy tale through the lens of microbiology.
Thank you very much.
Yours.
Once upon a time, there was a poor miller who had a beautiful young daughter. Now it happened that he got into conversation with the king, and to make an impression on him, he said, "I have a daughter who can spin straw into gold." The king was really impressed, and he said, "If your daughter is as skillful as you say, then bring her to my castle, and I will put her to the test." When the girl arrived at the castle, the king led her into a room that was entirely filled with straw. And giving her a spinning wheel, he said, "Get to work now. Spin all night long, and if by tomorrow you have not spun the straw into gold, you will have to die." This was the beginning of my favorite fairy tale when I was a child. I wanted to hear Rumpelstiltskin over and over and over again. Every night, the same fairy tale. So apparently there was something about this particular fairy tale which absolutely fascinated me. And probably you know how the fairy tale continued. So the girl was sitting in the room, terribly crying because she was not able to spin straw into gold. And then a little man entered the room, and he had the ability, but he wanted something in return. And after giving her necklace the first time and the ring the second time, the third time she had nothing more to offer. So she promised to give her firstborn child to the little man. Because now, after the third time, she would become queen. And so it should happen. The little man spun the straw into gold, the girl became the queen, and after a few years, she gave birth to her first child. And then the little man appeared again. "You promised to give me your first child," said the little man, but of course, the queen refused. And when I was a child, this was a pretty confusing situation for me, because I already learned you should not neglect the promise, but of course you should also not promise your child to a strange-looking little man, so... But there was no other solution to this. There was no other option to produce the gold for the king. But only a few years later, I was proved wrong. I grew up, I became a scientist, a microbiologist, and then, coincidentally, I stumbled across an article about this bacterium here, Cupriavidus metallidurans. And just by reading the first lines of this article, I immediately recognized that microbiology offered the solution to my childhood fairy tale trauma. Because this bacterium is able to convert toxic waste into gold and to accumulate them as form of gold nuggets in the outer membrane. So just with a little investment into bioreactors, process engineering, and upscaling, everything could have happened in a different way. So just imagine for a moment the fairy tale would go like this. The king led the daughter into a room that was entirely filled with bioreactors containing toxic waste. Giving her starting culture of the bacterium Cupriavidus metallidurans, he said, "Get to work now, ferment all night long, and if by tomorrow you have not produced any gold nuggets, you will not get a permanent position." But actually, this bacterium could also play a lead role in another Grimm's fairy tale. Because only in the beginning of this year, researchers decrypted the mechanism by which the bacterium produces these gold nuggets. And it just converts the toxic, soluble form of the gold into the non-toxic, solid form. And then it transports it through the membrane. Or simply speaking, it poops gold. Like a little micrometer gold donkey in the Grimm's fairy tale, The Wishing Table. But when I became a PhD student, I wanted to create my own fairy tale. So like every PhD student, I started my project with an exhaustive literature research. And if you're a bit familiar with the structure of fairy tales, you know that every good fairy tale requires a hero. Somebody who finds out the name of the little man dancing around the fire in the wood. Somebody who cuts open the stomach of the wolf to release the young goats. Or somebody who pushes the witch into the oven to prevent the brother from being burned to death. And actually, it didn't take me long to figure out who will become my microbial fairy tale hero. Because I already found it in almost every illustration in these fairy tale books. And also here in this illustration of the Red Riding Hood. But how can one see microorganisms in these pictures? Because usually they are too small to be visible to the naked eye. But there's one exception, and this exception are fungi. Because fungi are microorganisms which assemble to form macroscopic structures like these mushrooms down there. But usually fungi live below the soil surface. They thrive through the soil with their thread-like thin hyphae, but actually they can form pretty huge networks. And the largest living organism on Earth is a fungus. And only from time to time, they show up on the surface if they want to reproduce. And sometimes they appear on the surface in really strange ways, like in these regular circles here. And there are many legends around these circles. In European language, their names often allude to supernatural origin. In German, for example, we call them witches' rings because they are believed to mark the site where the witches dance on Walpurgisnacht. And usually, European superstition warn against entering those circles because in France, they are believed to be protected by giant toads that curse the ones who violate these circles. And in other parts of Europe, entering those circles would even result in the loss of an eye. But now that we had our microbial fairy tale hero. This makes only sense, of course, if there's somebody to be protected. And in fairy tales, these are usually young, beautiful girls, like Cinderella, like Rapunzel, or like Sleeping Beauty. The beautiful young princess who was mesmerized by an evil fairy to prick her finger on a spindle and to fall into a deep sleep from which she can only be awakened by a kiss from a king's son. And actually, I found a Sleeping Beauty bacterium, which is Bacillus subtilis. Bacillus subtilis is a soil bacterium. It usually lives close to plant roots, but it's also a sensitive soil. Because if the environmental conditions turn bad, it just holds up into such little spores, which are sleeping, and which will only wake up again if there's improvement inside. And in this context, I have to make a personal confession. So I confess myself guilty of torturing little bacteria until with their last ounce of strength, they released the endospores and died. The good news is that they weren't really dead, because these little spores, they are just sleeping copies of the larger cell. And in this sleepy stage, they can much better resist the bad environmental conditions I intentionally applied on them. The problem about this dormancy, as we call it scientifically, is that these sleeping bacteria are inactive. And in this inactive stage, they cannot provide all these important functions for us as they usually do, like protecting the plants from pathogens, as we heard in one of the talks before, or to decompose all the waste that accumulates into the environment, or to detoxify chemicals that enter there due to human activities. So what I wanted to find out was if probably fungi could help bacteria to regain activity. So what I did is I brought both together, and I waited some time, and after this time, nothing happened. But as a scientist, you have to learn to be patient, not directly throw in the towel, so I waited some more time, and indeed, after some time, the spores woke up. But for scientific curiosity, I wanted to find out what's the mechanism behind this regain in activity. Neither fungi nor bacteria have lips. It's pretty unlikely that a kiss is the underlying mechanism. And to test this, I used a small trick. So I provided labeled food and drinks to the fungus because I wanted to find out whether it shares it with the bacteria. And to measure the sharing, it does not need much. Just a tiny, tiny amount of these labeled compounds and a 3.2 million euro expensive super microscope that weighs more than 2 tons. But using the super microscope, we could show that indeed the fungus happily shares all its resources and that the bacteria were pretty hungry and thirsty. So they grow, they became larger and larger and larger, and suddenly they also reproduce. The positive side effect about this regain in activity and the newly generated offspring was the castle became clean again, so now they can fulfill again all of their functions, they can decompose the waste and detoxify the chemicals. And indeed, this would have been the perfect result to stop the story here with the magic closing words by which every tale finishes and they lived happily ever after. But as a scientist, it's also my duty to tell you the truth. I forgot to stop this experiment. And after a few weeks, I found the petri dishes again. They looked a bit strange. They also smelled a bit bad. So I put them again under the microscope. And what I saw there was everything else than a happy ending. So when the resources went scarce in this experiment, the bacteria started to attack the fungus and to feed on its biomass like little vampires. But there's no reason to be sad. Fungi and bacteria, they share our planet since millions of years. And neither fungi nor bacteria died out. Instead, they evolved many different types of interactions. Sometimes they fight each other off, sometimes they support each other, and sometimes this association is even so close that we can find bacteria living inside fungi. And even though my microbial fairy tale had no direct happy ending for the fungus, except the clean castle, it at least had a happy ending for me last year. And now, as a postdoc, I can continue my search for new real-life fairy tale examples by looking at them through the lens of microbiology. And I'm pretty curious to see which surprise awaits me next time. Thank you very much.
Very nice fairy tales, Anja, thanks. But it seems that we need to really treat with care our microorganisms, otherwise they will get some revenge.
Yeah, definitely.
Yeah.
We have to.
I have this fear. Yeah.
Our last but not least speaker is a professor at Lausanne University and a well-known microbiologist. Please welcome Jan Rolaf van der Meer. Jan, I also found a YouTube video of you with you about synthetic biology with almost 13,000 clicks, which is already something I would say for microbiologists.
Yeah,
so how
really
does it feel to
impressive
be
for
a
a
YouTube star, scientific YouTube star?
Well, you know, you have to compete with all these people that tweet, so you can either play on YouTube or you tweet.
And you chose YouTube.
Apparently.
Seemed like a good choice. Looking forward for your talk, Microbial Committee and Engineering. The stage is yours.
Thank you very much. So, dear audience, my title is Microbes and Us. All resistance is useless. We live with microbes. We cannot avoid them. We have to understand them. I asked my daughter to make a nice drawing of this, and I asked her to make a high five with microbes. She said, sorry, I can't. So now I will ask you to do a high five with your neighbor. And then we can also confuse the results from Josh. Thank you very much. And all the people on Livestream, please, in your home, also do the high five with your neighbors. United we stand, as I say. We live on a planet with bacteria that is dominated by bacteria, that is ruled by bacteria. The only possible way we have to favorably live with them is if we understand what they're doing. You've heard a lot about microbes, but then again, you have heard nothing about microbes. And I will tell you some of the secrets behind microbes. Let's first take a look at some of the pictures of microbes. Here's some beautiful cells, all different microbial cells that live in these things that we culture. Actually, we call them wastewater treatment plants. They're very useful because they eliminate all the wastes, hopefully, all the waste that we produce in our societies, that you flush down the toilet, everything ends up in the wastewater treatment plant. But in fact, it is the largest open source cultivation of microbes on our planet. It's a wonderful source for microbiologists to do these things. In fact, when you look at it, these bacteria do not live alone. They live in what we call communities. We have a society meeting here, so 2,000 scientists, they deliberate on communities of microbes, and they are totally enthusiastic about it. You can take microscopy images like this, and you can appreciate all kinds of things that grow together, and you can stain their DNA and then look at the DNA in their cells and appreciate the forms and so on. And you can go further and then see that there's hundreds of different species. And that is what the microbial ecologists love, you know. Oh, we can name them like this, and we can name them like that, and we're all very happy. We can make things very complicated, in fact. So many of the things that we do, and that's the summary of the whole conference that is going on for a week, is who is there? Like, oh, this cell, how do we call it? oh, how this cell? I found it. It looks the same, but I call it differently. It's very different. The second thing that we try to do is, what are these cells doing? And then the third thing, if they all live together, how do these communities function? And then the fourth thing that becomes very important also for our future is because if we have to live together with all these microbes that are on our skin or in the permafrost or everywhere, then maybe we have to learn how we have to live together, and if it's not perfect, how we can influence actually something. I don't like the word control because you cannot control microbes. Now I'm going to share you a secret. It took me 25 years to develop this secret, and it's the basic of microbiology. If you know this lesson, you know everything. The bacteria's quest in life, how to become two. This is a bacterium. All it wants, all it does, become two. How can it do this? Because it lives with food. What's going on with you people? A bacterial cell needs food to make two, and they're very good at this. In fact, they have been working 3 billion years on this problem. Unfortunately, bacterium, you're not alone. You're with many other species, you're with many other bacteria that try to do the same thing. Now, there's something curious about this food. It can be food like in a wastewater treatment plant, all the stuff we throw in. In some cases, the food is our body. We think of them as pathogens, but the bacterium thinks very different. It thinks, wow, there's a lot of food over there. How can I get access to it? Or if it's a bacterium that we call a plant pathogen that makes plant disease, the quest of the bacterium is, wow, now there's a lot of plant food over there. How can I get to this? That is all that the bacteria are thinking, and we have to understand and we have to appreciate it. The second lesson from this is, a bacterial cell is so tiny that the amount of food that one cell needs to become two is also very tiny. And that is why on the keyboard of your computer there's so many bacteria, because if you press your finger, you leave a fingerprint easily of a milligram of some fat, and that can produce 10 to the 9 bacterial cells, because one bacterium is maybe only 0.2 picogram of carbon, okay? Now, as I said, bacteria are not alone, they interact with each other. That's also very important to understand, because they all fight for their food because they want to become 2, and then 2 want to become 4, and 4 want to become 8. So we understand different ways that they can interact. For example, competition. There's some, you know, the guys and the girls that like a bit of fighting over there. Sometimes they cooperate, so they shake hands and they do things. I will explain that in a while. In some cases, they're just indifferent. I don't care who you are. Just go your business, I do my business, and we live happily afterwards. So take a look at this, few of these things, this competition. Imagine this. A bacterium is here. It lives in a solution of a sugar. A bacterium doesn't have a mouth, nor a nose, nor ears and eyes and hands and so on. How does it eat? Well, it has sort of a couple of straws, and it sucks the things in. So it puts this straw outside. It sucks all the nice sugars inside, and then this little machine that makes up the bacterial cell starts to work and starts to make 2. And they're happy. And they have used up all the food. But since they're not alone, this can happen. So here's our bacterium. It puts out the straws, tries to slurp. But there's another bacterium there that has many more straws. It slurps much faster. So as a consequence, they grow and this one doesn't grow. Now that's not a proper business. So that is where the bacteria really become a little angry and start to produce things to actually do something about this competition. For example, they can produce toxic substances, like to try and kill somebody else. Or sometimes they produce nasty needles, so stab them in the back, you know. You don't get a chance. If I'm not fast enough in straws and sucking in, then at least I puncture you to death. That's how it works. And that's what microbial ecologists like to investigate. But sometimes bacteria are more gentle, so then we talk about cooperation. So imagine you're a bacterium and you like plants. You have a problem, because a plant is not a sugar. A plant is something long, stiff, you know, they have polymers of sugars. You can put your straw out, nothing happens. You don't get anything inside, you cannot grow. So you have to invent a trick. Bacteria that invent this trick, what does it do? It first pushes something out, like a small scissor. And it's going to cut all these things to pieces to have this single sugar again that it can slurp inside, grow, and be happy and become two. And this leads to a lot of opportunities for other bacteria. For example, if you are a slurper for this long molecule, you send something outside, maybe this molecule that you liberate, there's another bacterium. So imagine one here and another one there. You produce one, I send out my straw, I can also profit. But because I'm gentle, I produce something that you need and so on. So you get like a cooperation. A lot of these things happen. And that's also what is done here at the ISME meeting. You know, tons of people, they try to understand how does that work, what do they send out, slurp, and so on, so forth. Sometimes it's even more interesting. So you can have cases where you actually have bacteria that like toxic compounds. So they slurp toxic compounds. It's ideal for us. We can clean up our environment. But bacteria, you know, despite all this evolution, there's some things that they cannot avoid. They are leaky to some extent. So they eat, but they lose stuff. And then there's others that sit next to me and they profit with their straws. And so you get all these nice bacteria growing. Then we call it a food web, okay? So that's cooperation. So there's all kinds of funny ways that bacteria can grow together. Now, where does this lead to? I said it's really important. We cannot fight microbes in that sense. We have to learn how to work with them together. So one of my own research interests is actually that we call that community engineering, that in cases where you have very badly contaminated sites, for example, that you help the microbes to clean up this stuff, to get going. So you could imagine that you have bottles with secret bacteria, like Alexandra was also showing you. You throw them out and magically they do their thing. And everything turns into... I show this because then I get some royalties from this brand of... You know, that happens to be a Swiss brand as well, but that doesn't matter. Now, just to show you some of the things that we like to do. So here's our magic bottle with bacteria. We throw it out on a site. And then we start looking at these things. We measure like all the things that the bacteria are doing. So for those who are not microbiologists, a bacterium has about 6,000 different genes, for example, so different functions that it can do. You can measure what they are doing in such an environment. And then we look at this picture and we say, geez, so that's about 6,000 different changes. How are we going to interpret this? That's the type of problems that we are facing. You know, we want to make that work, and you have this result, and you look at 6,000 different things. Or, for example, we look under the microscope, we have our favorite bacterium, it shares the food with others, we see that it works, and then sometimes it does very crazy things. Here, it is not sharing food, but it is sharing genes to others that then start to grow suddenly. So it's not just slurping with your straw, but you donate something else as well. Or here, you know, you do your best, you do your best, you grow here and so, and oops, now it doesn't work anymore, this video. Never mind. Yes. Can I get this working again or what? So these are growing, and here you do your best to actually degrade a toxic compound, you help the others, and then comes here a bugger and it eats you, you know? So these are the kind of unexpected things that we like to understand to make this thing working, that in case of complex communities, you can sort of steer them in a direction that you want to use. And that requires a lot of research, and that is why we need a lot of money as well. So, important lesson, huh? So, I don't like important lessons, but we should be very, very humble. We only start to begin to learn how microbial communities are actually working. It's embarrassing. So many scientists, they do all these kind of things, and if you look down to it, there's very, very little that we understand. We are beginning to learn how we can redirect these communities that live everywhere if we think that they are damaged. And we are beginning to learn how to build them even. If we want to achieve some process, we can try to build them by putting those microbes together on the basis of how they cooperate or not. Importantly, you can't beat the microbes. We cannot beat them. If we are not united with them, we will fail or fall. What is the correct? I have to ask the UK ones. Is it united we stand and then divided we fall, or we fall? Thank you very much.
Thank you very much. And this was the night of the microbes, ladies and gentlemen. I hope you really enjoyed such so fantastic and amazing talks that we had from our speakers, and that you also realize that those microbiologists and microbial ecologists have a sort of sense of humor, so to say. Huh?
Exactly. And that from now on, you don't change to the other side of the street if you see a microbiologist at your horizon. Yeah? He will be friendly. So may I ask all the speakers back again on the stage, please?
Yeah.
So Karen, Josh, Jan, Alex, we have some gifts for you.
A little gift for you.
Yeah, wait a sec. So just for you to know, we have a bottle of wine for the speakers, good bottle of wine, and the desperately desired you've set cup. So you're really lucky that you get one. So that was the night of the microbes. The bar will be open for another 2 hours, so you have the chance to get further refreshments and get in touch with the speakers maybe. So thanks for being here and get home safe.
Many thanks, yeah.
Bye.
Enjoy the